Hello, I'm Haley Branch, and I am a PhD candidate in Amy Engert's lab at the University of British Columbia. And I'm excited to share with you today some of the work that I have been doing on the effects of drought on leaf traits in scarlet monkeyflower. I will first provide a bit of background of what inspired this project, as well as my questions and the methods I used. Then I will share predictions for the traits of interest and the results, as well as a small discussion to tie these various traits together. And lastly, I will conclude with why all of this matters. So recently, there was a severe drought in California between 2012 and 2015, with the peak of that drought in July 2014. While California is known to experience cycles in wet and dry years, this particular event represented a mega drought. Robes in 2015 explored this megadrought by analyzing tree ring data from the past 1,200 years, which is represented in this graph, where the dotted red line shows the Palmer Drought Severity Index, which was approximately -20 for this 4-year drought period, a drought that has not been experienced by any other 4-year period in recent geological time. Since extreme weather events are predicted to become increasingly more severe and more frequent, I became interested in how plants could adapt to these unprecedented perturbations. Two of the specific ways that plants can evolve in response to drought is either through escape or avoidance. Traits that promote escape, or the live fast, die young method, are characteristic of growing fast and reproducing quickly prior to the onset of the more extreme drought period. Whereas drought avoidance traits, or as I like to call it, don't panic, grow slow traits, are morphological modifications that promote water stress but reduce water stress, but allow plants to still acquire resources. It likely doesn't come as a shock to any of you that the Engert lab loves Mimulus, but luckily it is also an excellent model organism for asking evolutionary ecology questions. So I specifically study Mimulus cardinalis, or scarlet monkeyflower, which is endemic to the west coast of North America, from southern Oregon to northern Baja California. And this encompasses the range of that recent severe drought. So fortunately, the Engert lab has studied this herbaceous species across time, collecting seeds throughout its range from generations prior to this drought and throughout this drought period as well. In this study, I will be addressing whether this species is able to respond rapidly to this severe drought event, and if so, through what type of adaptations, those that promote escape or those that promote avoidance. I set out to compare leaf traits from seeds collected prior to the drought and from those collected at the peak of the drought, using a resurrection method. In this approach, different generations can be grown and compared at the same time within a controlled environment, allowing for direct comparisons across generations. I planted these seeds in a controlled grow room and placed them in either wet or dry treatments. To compare leaf epidermal traits, I made peels with nail polish and observed these with light microscopy. To explore differences in leaf thickness, I hand-sectioned leaves and imaged them with bright field microscopy. And lastly, to compare physiological aspects of the leaf, I performed carbon assimilation response curves and compared them across the plants. The 5 ecologically relevant leaf traits that I will be exploring today are leaf thickness, stomatal density, guard cell size, trichome density, and carboxylation rate. For now, I will focus on the first 4. For the next section, for each trait, I will first provide a prediction and then the result. So firstly, I examined leaf thickness because I wanted to know whether shifts in macroleaf traits, such as SLA or specific leaf area, that are often used in evolutionary studies, were really because of anatomical changes. So plants that escape drought are expected to have thin leaves to maximize carbon gain and growth, whereas plants that avoid drought would have thicker leaves to resist gas exchange and increase water use efficiency. We found a significant increase in leaf thickness in plants from seeds collected during the peak of the drought, regardless of treatment. So for every figure that I present, I will have the trait of interest on the y-axis, and on the x-axis, I will have pre-drought and peak drought in that order. So as you can see, there was an increase in leaf thickness, and this was significant, indicating an evolution towards drought avoidance. Secondly, we looked at stomatal density, where stomatal density increased if which increase in stomatal density would correspond to drought escape, allowing plants to assimilate carbon and reproduce quickly. In contrast, fewer stomata would indicate reduced gas exchange and therefore drought avoidance. We found that fewer stomata were produced in general for plants during the peak of the drought compared to the pre-drought generation. As per the figure in the last slide, the trait is on the y-axis, but in addition to the generation on the x-axis, there's also treatment, where red corresponds to the dry treatment and blue corresponds to the wet treatment. So as you can see, although plants evolved fewer stomata overall, there was also a plastic response that was maintained, where dry treatments have increased stomatal density. In general, we would suspect a plastic escape mechanism here. However, I'm going to argue that this is avoidance, and that is because we also looked at guard cell size. If plants avoid drought stress, we expect that guard cell size will be smaller to reduce water loss and increase response rate. In comparison, larger guard cells are indicative of drought escape. We find an evolution in the direction of plasticity in guard cell size, which is really cool. So in the pre-drought generation, there's smaller guard cells in the wet treatment compared to the dry treatment. In contrast, the peak drought generation shows the opposite, with smaller guard cell size in the dry treatment. So this clearly suggests an evolution of guard cell size to promote drought avoidance. The last morphological trait we assessed was trichome density, where escape plants we expect would have fewer trichomes to allow for greater gas exchange, while drought avoidance plants would have more trichomes as they increase the boundary layer resistance, resulting in reduced water loss and increased carbon gain. Contrary to what we expected, we found no evolution in trichome density. There was, however, a plastic response where trichomes increased in the dry treatments compared to the wet treatments, and this was maintained across generations. So this indicates that this trait promotes plastic avoidance. So now I quickly want to tie in these four traits together. So there was increased leaf thickness, decreased stomatal density, and a switch in guard cell size plasticity, and all of these directly relate to the pathway for water escape. Additionally, the plastic response of trichome density also relates to gas exchange pathway. But why is this functionally important for the plant? This brings me back to the fifth ecologically relevant leaf trait, which I looked at, which is carboxylation rate. By measuring the CO2 assimilation response rate, we find that homeostasis is maintained, meaning that pre-drought generation is just as efficient in assimilating carbon as the peak drought generation. These assimilation rates were also maintained in dry treatments. Firstly, you may be wondering if my treatments were truly representative of a wet and dry treatment. So I've placed an example image on the slide of two siblings at the same developmental stage from either the wet or the dry treatment. You can clearly see morphological differences, although they're at the same stage. So the lack of relationship observed here in this figure indicates that there may not only be traits that promote avoidance, but there are likely also biochemical processes underlying that allow for drought tolerance of the plant. Now you may be wondering how exactly these morphological leaf traits that I measured relate to carboxylation rate. So let me tell you. My results suggest that the evolution and the plasticity of these four morphological traits are allowing for carboxylation rate to be maintained. So to summarize my findings in relation to carbon gain and water loss, I have created this schematic where the thickness of the arrows corresponds to how easily gas can move in and out of the leaf. So firstly, in the pre-drought generation, under wet conditions, plants lose a lot of water per CO2 gained. But this is okay because they aren't water limited. When these same plants grow under water stress, there are more stomata and of larger size, increasing the amount of gas exchange possible. The trichome density that increases allows for humidity, which is represented by this blue cloud, to build around the leaf, reducing water loss and allowing CO2 in. But there's still a lot of water loss per CO2 gained. The peak drought generation, when not water limited, has a thicker leaf and fewer stomata, creating a longer pathway of resistance for water to leave the leaf as well as for CO2 to enter. Now, when this peak drought generation experiences dry conditions, the thicker leaf is maintained, stomatal density increases, but the guard cell size is reduced. And this is key because the smaller pore size of the stomata now allows for better targeted CO2 assimilation, particularly with the reduced vapor pressure deficit, which prevents water loss. So thus, the peak drought plants under dry conditions have greater water use efficiency than the pre-drought generation under dry conditions. My next steps are to examine the internal leaf architecture to better understand the pathway of gas conductance once within the mesophyll cell layers. With that, I would like to thank all of my collaborators and those that helped gather the data for this project, as well as the bioimaging facility at UBC, where the microscopy was performed, as well as my funding sources, NSERC and the UBC Botany Department. Thank you so much, and I'm excited to take your questions.